So welcome everybody to today's co-hosted Qlucore Nabion webinar on the topic of exploring chronic liver disease gene expression data. My name is Hind Hashwa and I'm a scientific project manager at Nabion. And my colleague Anne will also introduce herself.
Hello, I'm Anne Holmberg from Qlucore and I do customer support, training and sales.
So the agenda for today's webinar is as follows. We will start with a short background about our solutions and the specific examples that we have chosen for today. And then we will have a use case demonstration live using our two softwares. We will end with a short question and answer session to address any ambiguities that arise. Feel free to use the chat throughout the webinar and we will address your questions at the end. So Qlucore Omics Explorer is the software provided by Qlucore, while Genevestigator is the solution provided by Nabion. And we will use both today in one common workflow. The two solutions have been designed to address some of the challenges that many researchers face. These are represented by the size and the complexity of public data. Our two solutions allow you to explore public transcriptomic datasets with visualizations and enriched annotations. In today's use case, we will use a dataset on chronic liver disease that has been enriched with annotations by the Nabion team and has been exported from Genevestigator into Qlucore using Qlucore's data format, GE data. Here we will explore, visualize, and analyze the dataset and the annotations, and we will use it to create a gene signature. We can then directly use this signature in Genevestigator for performing a compendium-wide analysis, which allows us to identify similar datasets and to also perform other types of analyses. Finally, we identify and export a dataset that we find interesting for further analysis in Qlucore Omics Explorer. We have chosen two studies from GEO, which use mouse models of NASH, with the titles and the accession numbers, which you can see at the top of the slide. NASH stands for non-alcoholic steatohepatitis, which is a very common liver disease that is a progressive form of non-alcoholic fatty liver disease. NASH is characterized by hepatic steatosis, inflammation, and fibrosis, and an optimal treatment has not been established yet. It can also progress into end-stage liver disease and liver cancer, such as hepatocellular carcinoma. Therefore, research using translational animal models of NASH is necessary for better diagnosis and effective treatment decisions. In these two datasets that we have chosen for today's webinar, the authors make use of two main mouse models of NASH without diabetes, which are established by feeding the mice a special high-fat and high-cholesterol diet, which we will refer to as CL. This is opposed to the normal diet. And they generate their data using an Affymetrix gene chip DNA microarray analysis conducted on liver tissue samples obtained from mice with identical genetic backgrounds. In one study, the CL diet is also deficient in methionine and choline, and hence we will refer to it as MCD. Additionally, in both studies, a group of the mice received the special diet together with a compound that is being investigated for the treatment of NASH. For instance, in the MCD diet study, the compound that is being used is called metformin, and this is an anti-diabetic drug for which the effects on inflammation and fibrosis had not been defined yet. Now, if we look at the sample numbers in more detail, we have 27 samples in total. In the first study, the CL diet, there are 5 mice that are fed control diet, 5 that are fed the CL diet and are hence diseased, and 5 that are also diseased but are additionally treated with a compound. And the same setting is applied in the second study, the MCD study, but here we have 4 mice in each group, and this makes 9 mice in total for the control group. So with that extensive introduction, I will hand over to Anne to start the live demo.
Thank you very much, Hint, for this very interesting background to the dataset. So I will now move over to the Qlucore software. Here we have it. And now we will start working with this NASH dataset that has been exported from Genevestigator. So the way to open that is to go here to File, Open. From here, we can also open a large number of other data types, such as RNA-seq, single cell, proteomics, metabolomics, and so on. But now we will do File, Open, find this GE data file, click on it, and that opens up the dataset, as you can see. And the dataset is always opened up as a PCA plot. And the PCA plot, which stands for Principal Component Analysis, is an excellent way of looking at your data in 3 dimensions. We have many more dimensions in the dataset. We have 27 samples and 20,000 genes. So we basically have 20,000 dimensions here. But we are only looking at the 3 first dimensions, which makes it possible to visualize it. And here we can see our samples. Each blue dot here is a sample. And we can see that the samples divide up in 3 groups. So let's try to understand what these groups are. So I go to the samples part here, and then I look at the annotations. So here you can see a long list of annotations that have been enriched by the Nebion team. And let's have a look at the diet formulation. As Hint told you, there are 2 diets and a group of normal controls. So let's have a look at that. Okay, so we can see that the groups are basically defined by the diets, even if there's also a difference here between the normal controls, and there is a big difference between the 2 different diets. Okay, that's interesting. Let's now open up a new synchronized plot and have a look at another annotation. And let's have a look at the stimulus annotation. Here we have information also about the treatment with the compounds that Hint mentioned. So here in the MCD group, we can see that the yellow ones are the untreated samples and the pink ones are the treated samples. And in the high cholesterol, high fat diet group, the red ones are the one without treatment and the green ones are the treated ones. Okay, so let's focus on the MCD diet for the moment and the corresponding control samples. Okay, so here we see again the 3 groups, the untreated, the treated, and the normal controls. And we see already that there is a difference here. We see that samples cluster together, meaning that they are similar to each other in each of the groups. And the distance between the groups indicates that there is a difference between them. So close together means similar, far apart means different. So that's a very easy principle to use when you are working with using PCA for visualization. Now I would like to find the best discriminating variables for the separation of these 3 groups. So I will do an F-test, an ANOVA, or as we call it, a multi-group comparison on the annotation stimulus. So I will now work with the 3 active groups. I have deactivated a couple of groups here that I'm not using for the moment. So by just dragging the statistical slider here, I will set the cutoff for the P and Q values. So with the Q value of 0.01, the false discovery rate or the corrected P value of 0.01, which is really good, I find 6,200 genes. Okay, and we see that for these, the expression of these genes makes the samples cluster even closer to each other, indicating that the expression for these genes within the sample groups are very similar. But I would like to have a shorter list, so I continue to filter down to somewhere below 1,000, so 800 or so. And now I would like to show these genes in a heat map. So I changed this plot now to a heat map. And I want to configure the heat map so that I see everything. I want to hierarchically cluster it like that. And I want to color it also according to stimulus. So I pick the stimulus annotation and color by that. And then we see the same groups in the heat map as we see in the PCA plot, and we also see that they cluster very well together in the heat map as well as in the PCA plot. And now I will flip two of the clusters here, change the order of them, which I'm allowed to do. Now I have the untreated MCD diet samples here, the treated ones here, and the normal controls here. And then we can see that within the treated group, we have a couple of genes which are becoming more red for the treated group here, indicating that they are becoming more normal after treatment. And we see the same tendency down here. These genes are becoming more blue, becoming more normal after treatment. So that's an interesting finding. And we can see a tendency also in the PCA plot, not as clear. So this is quite a weak signal when you are looking at so many genes. Okay, and now maybe we would like to open up a new synchronized plot and have a look at a box plot. So let's have a look at some of the best genes here. So here we have a long list of all the plots that we support, and from here we pick Boxplot. And the Boxplot I need to configure, so I need to decide what goes on the x-axis. I would like to have stimulus there, and then I need to decide which genes I want to show. And I do that from the Variables tab. I now pick the full list of 820, and that's too many to show. So let's add some information to the list here and sort on Q values so that I get the best genes first in the list here. So now I can pick one or many genes here. So now I show the 4 best genes here. You can see that they are very good, at least at separating normal from MCD. Let's have a closer look at one of them like that. Let's change the label to gene symbol like that. Let's close this window. And let's also configure it a little bit so that we show the individual samples. Okay, and here we can also show the p-values for the combination, for the separation of each of the pairwise combination of the groups. Okay, and then you can step down if you just want to scroll down through a lot of different ones, or maybe if you want to pick a number, you can just pick a number like that. And this can, of course, Boxplot can easily be exported by just right-clicking here, export image, or even copy it to clipboard. Okay. Next step here now is that I would like to generate a gene signature that I will export to Genevestigator. So let's close down the Boxplot for the moment, tile these plots, and now I want to focus on genes that are good at separating the MCD untreated group from the control group. So let's take away the treated group for the moment like that. And then I want to do a t-test. We call that a 2-group comparison. And here I want to compare stimulus, the MCD group, with the rest of the active samples. So right now the blue group is the only, the control group is the only remaining group. So then I will compare the MCD group with the normal controls. And then I will just again move the statistical slider to a good Q value. So with a Q value of 0.01 again, I find 5,000 genes. That's a lot. And I have them, okay, I don't see them in the heat map yet, but I want a shorter list. So I the gene signature that I want to export to Genevestigator, I want that to be around 50 genes or so. So let's just try to achieve that. Sorry. Like that. So 57 genes, and I can also have a look at them in the heat map here if I want to, and I can of course change the label for on the y-axis here so that I see the genes. Okay, so these 57 genes I now have here in my active list, and I can make a copy of that and call that MCD versus control. Like that. Maybe I would like to add some more information to the list. Maybe I would like to have the p-value. Maybe I would like to have the fold change, and maybe also the log fold change, which we call the difference. And this is actually the value that I will export to the Investigator. So now and I can export this list by using this icon here. It will be exported as a TXT file. But now I want to export it to Investigator, and then we have a quick path. So by just choosing copy here, I can then move over to Investigator, which is a separate software that was already open here on my computer, and I will here select signature, the signature tool, and I will enter the signature. So by just pasting here now, like that, I paste the signature that I just copied, and by that I leave over to Hint, who will now tell you what you can do with this gene signature in Genevestigator. Over to you, Hint.
Thanks, Anne.
And I'm now making you the presenter.
Correct. So you should be able to see my screen with Genevestigator open.
Yes.
Great. So I will use the exciting signature that Anne has created in Genevestigator that was defined based on differences in expression between the MCD-fed and the control diet-fed mice. So for the analysis that we will perform in Genevestigator, we will address two main questions. The first is, can we verify whether our signature is really specific for this disease? And the second is, can we validate disease biomarkers? So to answer the first question, we need to define a data set that includes other published studies, which we can use to compare to our results. So in Genevestigator, I have already defined a data set, including experiments from mouse that we have on the Affymetrix platform, which is the largest resource for microarray data. You can see this here on the left. It contains multiple experiments with multiple individual samples, making a total of over 12,000 samples. Since we are interested in the signature that we have received and we would like to visualize these genes in the selected datasets, we can go directly into the signature tool down here, which is what Anne has already shown you, to enter our signature. I will click on Enter Signature and simply copy-paste the list that I have received. I need to then define the scaling of the data. So this is in log2 format, which is called differences in the Qlucore software. And to define that the data is relative, because it was defined using a comparison between the diseased and the control mice. And I can simply give this a name. So this was the MCD versus control, and I will click on OK. And before I start my analysis, I need to confirm the list of genes that correspond to the entered identifiers. So when I click on OK, the signature appears. So I have several genes that have a red color. They are upregulated, and ones in green, which are downregulated. So we have a combination of both. And we have set the tool to identify the most similar profile to the one that we have inputted. We will make use of the Pearson correlation as a distance measure, and then we can simply click Run. So the results that we get are based on the values of expression. For every row that you see here is a comparison between two different groups of samples, which we call perturbations. And every column that you see here is one of the genes that we provided in our list of genes. As you can see, many of the rows that are identified are actually comparisons between NASH, steatosis, or liver fibrosis compared to control liver tissues. You can see more information by moving this to the right. And you can get more information about each by simply placing your tooltip on top, and a window will appear which contains more information about that study. Since we see many NASH-related dietary models, it means that we could identify several comparison sets that give a similar response to the dataset that we previously explored in Qlucore's Omics Explorer. So this represents one way to verify your results, because you can identify several other studies than your own dataset in which a similar pattern of differential expression for a set of genes exists. Now, if we look in more details at the list, we can identify several other comparison sets that are interesting. They represent other diseases. For instance, there are liver aging studies comparing between two different time points. There are treatments with certain drugs. For instance, the one you see down here is a sodium arsenite study, which investigates lung tissue and not liver. And there are also two studies on liver cancer, hepatocellular carcinoma, such as the two you see here. So you see, this approach helps you to identify not only other studies which confirm your signature in the disease that you are interested in, but it also allows you to look at samples from other organs, cell types, and diseases, and can bring you a lot of information starting from a single signature. So this one hepatocellular carcinoma study, I will record the identifier and come back to it at the end of our demonstration for further exploration in Qlucore's Omics Explorer. Now, the signature that we used was based on the difference between MCD and control mice. But you might recall from the introduction that the experimentals also treated mice that had been fed the MCD diet with a compound, metformin, which they wanted to investigate as a possible therapy for NASH. So if we are interested to see how do the samples that are treated with this compound look in terms of the expression of this selected list of genes, we can structure the comparisons further and look for clusters of similar genes and similar samples in terms of expression. And for that, I will simply move over to the hierarchical clustering tool, which you see up here. So now I have the same list of genes that I received from Anne, but I would like to define a new data selection, which contains only the samples from the original MCD study. For that, I will make use of the search by accession number, and I will simply type in the number of the study that corresponds to the MCD. The identifier that I'm using here is the Genevestigator ID. Of course, one can also make use of other IDs such as GEO. So this is the MCD study. I give it a name. And when I click OK, it will load here as a new data selection containing only one experiment with the 12 samples. So we would now like to choose the samples from the drop-down menu as our second biological dimension. And we will make use of the Pearson correlation to cluster both genes and conditions. When I click on Run, this clustering is completed. So what we see here on every row is a sample instead of the comparison that we saw in the other tool. And on every column, we see one of the 57 genes. And when looking through the results, we notice that the biological replicates seem to cluster very nicely together. So every 4 biological replicates seem to have very similar expression, and the MCD metformin treated group seems to cluster more closely to the MCD group, and they also have a more similar pattern of expression compared to the control. This is also what Anne observed in the heat map visualization in Qlucore Omics Explorer. So coming to the second question that we posed, could we use this information to identify two genes that are strongly differentially expressed between the disease and the control, which would allow for a better visualization of the compactness of the cluster? So I chose two genes which show a strong and different expression between the sample groups. I will make use of this UBD and the MBL2. And I can simply do that by unticking the list of genes and then reselecting only the two that I'm interested in, like that. So now you see why I chose these two genes. They seem to have medium expression in the metformin treated group, but either really high or really low expression in the control and the NASH the NASH untreated groups. And now I can jump over into the two gene plot since I defined only two genes that I'm interested in. And this scatter plot now shows me the LOC2 expression of the two genes across all the different samples of my data selection, because the samples is chosen up here from the drop-down menu. And I can see that the samples cluster. I can use the stimulus to give each of these groups a color. And this will verify again what Anne has seen, that the stimulus seems to separate the groups really nicely, and that the metformin treated group seems to be moving away from the MCD towards the control. And here, for instance, is one potential biomarker of NASH that can be verified further, of course, experimentally. If we would like to export this figure, we can simply go up to Results, Export Image in different formats. There's a lot more analysis that one can still do in Genevestigator, but what we would like to finish with is to bring one of the studies that we identified using the signature tool back into Qlucore for further detailed statistical analysis. So I will take the example from before, the hepatocellular carcinoma study, and add it as a new data selection, again using the Genevestigator ID. One can again use here the other accession numbers. So this is the number that I recorded, and when I search for it, it will find the original hepatocellular carcinoma study that we found interesting. So I will load it like that, and it will appear as a new data selection with 39 samples. We are curious to quickly take a look at what these samples are. We can go back to the Home button and make use of the perturbation tool under the condition search tools and change from a scatter plot list to a tree view to visualize that we have multiple samples from a NASH group of mice, but also from liver neoplasm. And here you can see the expression of the two genes of interest, which can be used for sorting, or we can simply click back the entire list of 58 genes and visualize these by using the heat map tree view. Now to explore this data set in detail, users of the Enterprise Edition can export the single experiment from Genevestigator and import it into Qlucore. For exporting, the button can be found under Datasets Current Selection, Qlucore. And it will be exported as a GE data file and can be opened directly in Qlucore. Since this step takes a few seconds, I've already loaded this data selection, as you can see here, and I can simply click on this icon and it will open up in the other software, so in Qlucore Omics Explorer, and you can see here the PCA plot with the different annotations that have been added by my team at Nabion. I can again use the stimulus to color these groups of samples. As you see here, we have some NASH groups and the hepatocellular carcinoma group. And I can continue exploring, performing statistical tests, similar to what Anne has shown you in the beginning of this webinar. So with that, the live part of the demo is now over, and I will hand over back to Anne for a short summary.
Yeah, hello again, and thank you, Hind, for this very impressive demo of what is possible in Genevestigator. And to summarize, we have shown you a data set on chronic liver disease enriched with annotations from Genevestigator. And we used Qlucore Omics Explorer to explore this data set, where we examined the different dietary NASH mouse models, and we did observe an effect of the diet composition and also an effect of treatment on the liver gene expression. And then we created a gene signature comparing MCD diet with control, and this gene signature was copied to Genevestigator using copy-paste. And then Hind showed us how to use this gene signature in Investigator. So she did a comparison across a whole compendium of public data sets and identified similar data sets. Then she also showed us how to identify biomarkers of NASH. And finally, she took one of the identified data sets and exported it back to Qlucore Omics Explorer, and thereby closing the loop, the analysis loop, so that we can then take this data set and start a deeper analysis of it in Qlucore Omics Explorer. Yes, and finally, the conclusions. So as you can see, the combinations of our two softwares provide an end-to-end solution for gene expression analysis. So with Qlucore Omics Explorer, you can perform deep statistical analysis of single experiments and visualize in many different plot types. And with Genevestigator, you can perform meta-profiling across thousands of samples from public studies. So this brings a big synergetic effect to your analysis. And if you are interested in trying either of the softwares yourself or the combination of them, you can download both of them from our home pages. And we will provide you with full support during your trial. And we will send out a follow-up email after this webinar with these download addresses as well. Okay, and by that, Hind and I would like to thank you very much for your attention today, and we open up for questions.